Hello viewers, my name is Mr. Mtiane from Motherwell High School in the Nelson Mandela District. Welcome to the Eastern Cape Department of Education broadcast studio. Today's lesson is about the DNA, the code of life. Firstly, we need to understand why is DNA regarded as the code of life. First of all, it contains the instructions that instruct how the life processes should happen in living organisms. The examples of life processes are protein synthesis, nutrition, excretion, how all those processes happen in the body of an organism that has been encoded in their DNA. A very useful document that we use in life sciences is the examination guideline. This examination guideline is showing you the examinable content of what we are studying in life sciences. So with regards to this topic, today I will focus on the location of the DNA, the structure of the DNA, and the functions of the DNA. This is an outline from the examination guideline. What are you required to know for the location of the DNA, and what are you required to know for the structure of the DNA, and also what are you required to know for the functions of the DNA. This is outlined in the examination guideline because it is examinable. The first thing that we are going to look at is the location of the DNA. When we look at the location of the DNA, we have to consult the historical question papers on how this question has been asked before. There are two ways in which this question has always been asked. They can say the location in a cell and a location in the nucleus. So a learner needs to know what to answer when the question speaks about the cell and also a question when it speaks about the nucleus. Let us start with the location in a cell. There are two types of eukaryotic cells, which is plant cell and animal cell. So let us start with a plant cell, the location of the DNA in a plant cell. There are three areas where you will find the DNA in a plant cell. These organelles, it is the nucleus, mitochondrion, and the chloroplast. But when we look in animal cells, we only find it in two of the organelles I've already mentioned, which is the nucleus and the mitochondrion, but not the chloroplast. So learners in an exam, they need to be mindful whether is it talking about a plant cell or whether is it talking about an animal cell. Even though sometimes it will not say animal cell, they will say human cell. A learner will need to have a judgment that a human is an animal, so they would know that they do not include chloroplast in their answers. But let us look when the question has been asked in a way that the location of the DNA in the nucleus. For example, the nucleus of a cell, it looks different when a cell is dividing and when a cell is not dividing. The nucleus looks different. Number one, if we speak of the location of the DNA in a non-dividing cell, we will look at the chromatin network. But that chromatin, as soon as the cell starts to divide, it changes into becoming chromosomes. So the two answers when a learner is asked in an exam about the location of the DNA in the nucleus, it is the chromatin network and as well as the chromosomes. So to sum it up, the location of the DNA can be looked in two ways, in a cell and also in the nucleus. In a cell, we look the plant cell, which has got three areas or organelles, which is nucleus, mitochondrion, and importantly, the chloroplast. But with animal cell, we just look at the nucleus and mitochondrion. But in the nucleus, we look at the chromatin network or the chromosomes, depending on whether the cell is dividing or it is not dividing. Let us proceed to the next part of the lesson, which is the brief history of the discovery of the DNA molecule. Who discovered the DNA molecule? For example, our examination guideline and the CAPS document that we use to teach, it has included 4 people that are involved in the discovery of the DNA or as well as the discovery of the structure of the DNA, but they have different inputs with all this discovery of the DNA. So sometimes in an exam, you can be asked in a particular aspect that it requires certain people from this group of 4 people that were involved in the discovery of the DNA. But let's look at the brief history. In 1952, Rosalind Franklin and her assistants, Maurice Wilkins, they researched on the structure of the DNA using X-ray diffraction images. Watson and Crick in America, they did an independent research of the DNA. But upon seeing Franklin's images, they then proposed a 3-dimensional double helix model of the DNA in 1953. Then in 1962, Watson and Crick, they received their Nobel Prize for the discovery of the DNA structure, and Wilkins received an award for the X-ray photography, while Rosalind Franklin, at that time, she was already deceased, and they do not give a Nobel Prize to the deceased. Let us look at these humans or people that discovered this structure of the DNA. These are the people. Here, in the first picture, we have Watson and Crick. On this side, we have Miss Rosalind Franklin and her assistants, which is Wilkins. So Rosalind was working with Wilkins in trying to discover the structure of the DNA, but Wilkins took the pictures and showed them to Watson and Crick, and Watson and Crick, they had that aha moment where they could now say, oh, this molecule actually looks like this, and then they proposed that 3-dimensional structure of the DNA. But they were all involved in the discovery of the DNA molecule, but they have different inputs in that. The examination guideline that we are using, it has cautioned us, the diagnostic report has cautioned us on how to answer some of these questions. For example, in the 2024 paper 2 examination, a question about the DNA discovery was there, and it was in question 1.3. And let us read what this diagnostic report found. It said that most candidates performed well in question 1.3. Some candidates struggled with the history of the discovery of the DNA, and that question was question 1.3.2, even though the scientists involved are specifically named in the examination guideline page 7 and as well as the CAPS document. The controversy regarding the earlier recognition of the scientists might have stumped the candidates. So the candidates are required to know who did what and what they did collectively as these scientists, because the question can come asking for what Watson and Crick did, what Franklin and Wilkins did, or what they all did. They all discovered the DNA molecule, but they did different things in the discovery of the DNA molecule. So from now on, as I sum up, learners need to know that all these 4 people are involved in the discovery of the DNA structure, but are involved differently, and they can read more of that in their textbooks. Now, let me proceed to the structure of this molecule, which is the DNA. Naturally, a DNA molecule is a double helix. I will explain that further on. I will show how it looks when we say it is a double helix. Now, it consists of monomers called nucleotides, and this here is a nucleotide. So this is a basic unit of a DNA molecule. When you assemble these monomers together, you might end up with a DNA molecule. Now, when we look at the monomers of the DNA, because a monomer of the DNA has got 3 parts itself. Number 1, it has got the phosphate group, a sugar molecule, and a nitrogenous base. So these are the 3 components of a monomer of a DNA, which is a component of a DNA. Now, when they are joined together, these monomers, they form long chains which are called the polymers. And in this case, the polymer is the DNA, the monomer is a nucleotide. The sugar in the DNA, this sugar here, it is called a deoxyribose sugar, and it is attached to the nitrogenous base on the side. And also the phosphate and the sugar, which is this part and this part, are attached to one another by the strong bonds. And this is a bond that is attaching a phosphate and the sugar, alternatively, to form a long chain. For example, this sugar here, it can be attached to another phosphate. And this phosphate up there can be attached to another sugar to form a long chain of this structure. Now, coming to the nitrogenous bases, because we have explained that the name of the sugar is deoxyribose sugar. For example, when the learners are asked in an exam, they need to specify it correctly because there are many sugars out there: glucose, ribose, pentose, maltose. There are many sugars that right out there. So they need to specify this one as deoxyribose. Now, with nitrogenous bases, we've got 4 groups of nitrogenous bases or types. We have the adenine, we have the thymine, we have the cytosine, and the guanine. If a molecule does not contain these all 4, then it is not a DNA molecule. For us to know that this is a DNA molecule, it needs to have all these 4 nitrogenous bases in it. They are very important. Now, here on the side, as we continue with the structure of the DNA molecule, we hear that these nitrogenous bases are complementary. They are complementary. In other words, they join together, but in a specific order. What do I mean by that? Adenine always joins with thymine. Guanine always joins with cytosine. As you can see here with the shapes, this shape can join here, and this shape can join here. So this is how these things happen. So as you can see yourself, this A cannot join to this C. And as well, this G cannot join to this T, which means that these bases are always complementary. And now let us proceed to the next slide on the structure of the DNA. This is how naturally a DNA looks like. This is the double helix I was talking about. It looks like this. And now let us look at a simplified version of this molecule. You look at it, it has got 2 strands. There is a strand on the left and there is a strand on the right. In other words, it has got 2 strands. Hence, it is regarded as a double-stranded molecule because it has got 2 strands. And these strands are joined to each other by the bonds which are called weak hydrogen bonds. And these weak hydrogen bonds, they exist between the nitrogenous bases that pair. Between adenine and thymine, there is a weak hydrogen bond. Between guanine and cytosine, there is a weak hydrogen bond. Now, as we conclude on the structure of the DNA molecule, learners need to know that first of all, it is a double helix, and it is a double-stranded molecule that has got these strands joined by the weak hydrogen bonds, and it has got the monomers of it, which are called the nucleotides. These are the monomers. For example, in this DNA that has been shown here in the diagram, we have got 10 monomers that make up the DNA molecule. Now, let us proceed to the functions of the DNA molecule. It is important to consult your examination guidelines because when you are asked about the functions of a DNA molecule, you are expected to take them from the examination guideline. Do not come up with your own functions of a molecule. These are the molecules that are written in the examination guideline. And it's not easy, it's not difficult to memorize these things. For example, the first function says the DNA makes up the genes which carry hereditary information. So that is the first function of the DNA, that it makes your genes which carry hereditary information. The hereditary information we are talking about, it is the genes themselves, which are passed from parents to their offspring. The genes I'm talking about, it might be the height, it might be the eye color, it might be the hair color, it might be your left-handedness, it might be anything that you have inherited from your parents. And lastly, it says that it contains a coded message for protein synthesis, or a coded information for the synthesis of proteins. So for proteins to be synthesized, first of all, there must be an instruction from the DNA that is telling on how these proteins are to be synthesized. So in essence, it contains a coded message for protein synthesis. Remember, in grade 10, you learned that proteins are large molecules. They cannot enter or pass through the cell membrane. So a cell needs to synthesize its own proteins from the amino acids of the proteins. Now, to sum it up, when it comes to the functions of the DNA molecule, you have to look no further than your examination guideline. That we have got two functions that are explained in the examination guideline, which is the DNA makes up the genes that carry hereditary information. The DNA consists of the coded information for protein synthesis. Bond W. The bond W, it is what we call a hydrogen bond. A hydrogen bond, it exists, or I do not need to write bond either way, but let me continue writing. So it exists between two nitrogenous bases. These are the nitrogenous bases. So between the nitrogenous bases, you will find a hydrogen bond. Now, question 1.4.2 says, give a collective name of parts X, Y, Z. Now let's look at our diagram. What is the collective name for X, Y, Z? So this is X, Y, Z here. So it's a three-part structure. It's a three-part molecule. And you know those three parts. And that is what we call a nucleotide. Let us write it here. It's called a nucleotide, which is a basic unit of the DNA molecule. And if you can look in the diagram above, we have about 4 nucleotides, because each nucleotide needs to be made up of a phosphate, sugar, and a nitrogenous base. So you find 4 of the nucleotides in the above molecule. Now, what is the natural shape of the DNA molecule? That is written in your examination guidelines. There is nothing that is important as your examination guideline in this subject. So the question says, state the natural shape of the DNA molecule. It's simple. The natural shape is the double helix. The natural shape is the double helix. That is the natural shape of a DNA molecule. And then we have question 144. It is something that we did not speak about in our lesson, which says, name the process whereby the DNA makes a copy of itself. And now you have to write that on the comment section. Answer this question on your comment section to show your research skills. Go and find out the process whereby the DNA makes a copy of itself and write your answer on the comment section. Now, the last question is very important because it is speaking about the location of the DNA, particularly in an animal cell. It's very important. If you remember, we said that in plant cell it is found in 3 places, and in animal cell it is found in 2 places. Now, what are those areas or places or organelles where you'll find the DNA in an animal cell? Now, the answer here, it is going to be the nucleus or the mitochondrion. So those are the 2 answers that are expected there, the nucleus and the mitochondrion. Let me write it here. Nucleus and mitochondrion. You don't have to write chloroplast in this case because chloroplast is not in animal cells. It's only in plant cell. And this is the end of the question. And we have come to the end of today's lesson. You can leave a comment or a question. Please do not forget to leave the name of your school so that we can do shout outs. And please share this and recommend and subscribe to our YouTube channel.